My name is Rebecca, and I will be your X-Talks host for today. Today's webinar will run for approximately 90 minutes. This presentation includes a Q&A session for our speakers. This webinar is designed to be interactive, so please feel free to submit questions and comments using the questions chat box. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode, and the presentation slides will advance automatically. Please note that this event will be recorded and made available for future download. We would now like to thank Spaulding Clinical Research, who has helped develop the content and presentation for today's meeting. Spaulding Clinical Research, LLC, a change agent in the clinical research and medical device industries, and process innovation that delivers speed, unprecedented quality, and lower costs. Spaulding Clinical Research operates a 105-bed clinical pharmacology unit with 96 12-lead ECG telemetry beds in West Bend, Wisconsin, USA. The facility is paperless, using a customized Phase 1 electronics data capture system with biodirectional interfaces to clinical lab, bedside devices, and telemetry. Spaulding Biometric provides full-service biostatistics data management and medical writing services. As a Phase 1 through 4 centralized cardiac safety provider, Spaulding Clinical offers project management, expert cardiologist ECG overread, and a complete suite of state-of-the-art equipment provisioning. This includes the Spaulding Web ECG Diagnostic 12-lead ECG management solution with the purpose-built Spaulding Model 1000 iQ'd electrocardiograph. At this point, I'd like to welcome our moderator for today, Mr. Daniel Selness. Mr. Selness, you may begin when ready.
Thank you, Rebecca. I'm very pleased to be here moderating today's webinar entitled Farewell to TQT Studies. I'm also very happy to welcome all of you on behalf of Spaulding Clinical, where I serve as the General Manager and Senior VP of Research. I've had several clients comment to me on what a surprising title this was for today's webinar, considering Spaulding's clinical expertise in thorough QT studies. While we are experts at conducting TQT studies, we actually do conduct all types of clinical pharmacology studies. And what we're focusing on today is discussing the growing trend for building your cardiac safety profile for your compound, starting with your first-in-human study. We have two great experts as presenters this morning, and I don't want to take any more of their valuable time, so let me start by introducing Dr. Jay Mason. Dr. Mason is a world-renowned cardiac safety drug expert with 35-plus years of experience in cardiac care and research. He graduated from Princeton and obtained his MD degree from the University of Pennsylvania. He also trained in medicine and cardiovascular diseases at Stanford University, where he was a member of the faculty from 1975 to 1983. He was chief of cardiology at the University of Utah from 1983 to 1999, where he still serves as a faculty member, and served as chairman of the Department of Medicine at the University of Kentucky from 1999 to 2003. Dr. Mason's clinical, teaching, and research emphasis is in cardiac arrhythmias and electrophysiology. He has authored over 400 publications and has served on the National Research Review Committee for the NIH and the American Heart Association. He also served on several editorial boards, including the American Journal of Cardiology, Circulation, Annals of Internal Medicine, American Journal of Medicine, and the Journal of American College of Cardiology. Dr. Mason is currently the Chief Medical Officer for Spaulding Clinical Research, and his topic today is cardiac surveillance strategies to improve safety and avoid the thorough QT study. Before he gets started, we have a couple of poll questions for the audience. So Rebecca, please present the first poll question.
Thank you, Mr. Selness. So our first question for today is, do you think it is necessary to perform some form of repolarization testing in humans on all or nearly all drugs? That is the arrhythmia problem common enough that the methods for detecting and preventing it effective enough to justify the requirement, the time required, and cost? And you have two options here. You can choose yes or no.
Rebecca, this is Jay Mason. Should the questions be appearing on the screen?
I believe they should. Sorry about that. Let me just make sure that they are launched.
They aren't on mine. Perhaps they are on others.
So it looks like we have a few who have, about 71% of you all have already voted. So I'll just repeat that question. Do you think it is necessary to perform some form of repolarization testing in humans on all or nearly all drugs? And you have two options. You can click yes or no. Okay, looks like almost all of you have submitted your answers, and I'll just share these now. So in answer to the question, do you think it is necessary to perform some form of repolarization testing in humans on all or nearly all drugs? 66% of you answered yes, while 34% of you mentioned no. And Dr. Mason, do you see these results now?
No, I don't. I don't see the poll or anything other than the chat box that I suppose others can't.
Why don't I go ahead and start off and launch our second poll and then poll question, and then we can talk privately about making sure that you're all set up for your presentation.
Okay.
Great. So as mentioned, we have a second poll question for you today, and let me just launch that right now. So our second poll question for this morning, do you think the E14TQTS is an appropriate method for preventing drug-induced TDPs? And again, you have two choices. You can click yes or no. Great. Thank you, everyone. Almost all of you voted today, and it seems like a straight 50-50 split. So in answer to the question, do you think E14-2QTS is an appropriate method for preventing drug-induced TDPs? 50% of you said yes, and 50% of you said no. And so our last poll question for this part of the presentation anyway,
What's the percentage
right now, and you can see it on your screen right now. So our third poll question for this morning, if there are no TQTS requirements, would your company likely bring more drugs to market? And again, you have two options here, yes or no. Thank you, everyone, for participating in our last poll question, which was, if there were no TQTS requirement, would your company likely bring more drugs to market? 42% of you said yes, while 58% of you mentioned no. So at this point, I'd like to pass the presentation over to Dr. Mason, and I'd like to hear his insights on what he thinks about what we gathered from this poll question.
Thanks, Rebecca. I wonder if we still have a problem. I'm sharing my screen, but I can't see anything other than the chat box. Can you see my screen?
Yes, Dr. Mason, we can see your screen. Would you mind just loading up your PowerPoint presentation?
I will do just that, and then I will tell you what I think about these answers.
Yeah, I think we're all dying to know. Great. So you've had those poll questions presented right on your screen for us to see here today, and I'll just let you take it from here.
Rebecca, is the chat box visible to you?
Nope, not at all.
Good.
All we can see right now is your PowerPoint presentation, as well as the toolbar with just some of the applications you have installed on your computer, like Microsoft Office and Adobe.
Okay, good. Very good. Well, the first question, the majority, 2/3, said yes, and I certainly think that that's appropriate. As we'll briefly discuss, there is a substantial incidence of arrhythmia problems due to QT prolongation, and obviously this needs to be addressed in some way. The second question is about what the response is about what I expected and what I believe. Half of you said that the E14 method for assessing repolarization liability is a good way to do it. That means that half of you don't like the E14 approach, and I must say that I don't think it's ideal. In fact, that's really the topic of this webinar. Number 3, would more drugs be developed? That's worrisome. 42% said yes, and that's a reason why both I and Dr. Benson and many others do feel strongly that the thorough QT study should be altered in some way so it is no longer an obstacle to drug development. Okay, let's go on to the, that's interesting, I'm not able to advance my slides. How about this way? There we go. Everyone should be seeing a table listing drugs that have been withdrawn from the market for QT prolongation. Rebecca, if you're not seeing that, let me know right away. The list is pretty long. There's 16 listed here. The problem starts in 1988, and the most recent withdrawal was the higher IV dose preparation of ondansetron. So it is a substantial problem. Here's what we're going to talk about during the next 25 minutes. I'll briefly review what the original E14 guidance required, and then I'll update you on new requirements and clarifications that have resulted from the various Q&As that have been issued by ICH, FDA, and Health Canada. And I'll add additional clarifications that routinely appear in FDA response letters to thorough QT study protocol submissions. Then I'm going to give you a list of what I consider to be the more important deficiencies of the current E14 approach, and I'll follow that by providing alternative strategies in each case. This is the original guidance. I'm sure that everyone is familiar with it, and I won't spend any time other than this slide to discuss what its requirements are. As you know, it applies to almost new drugs, almost all new drugs, and roughly 95+% of new drugs, in fact, are having to undergo this requirement. The requirement is to detect a 5 millisecond change in QTc with an upper common boundary of 10 milliseconds. I won't get into the statistical implications of this beyond indicating that this means you have to do a lot of ECGs in a fairly large study. The number of ECGs in studies I've been associated with has ranged from 4,000 to 30,000 ECGs. The E14 requires that ECG interpretation be centralized, and the centralization should also include uniformity of equipment. The study must have a placebo control and an active control, which is usually moxifloxacin. And whenever possible, it should explore a supratherapeutic dose, which ideally should be at least 4 times and up to 10 times the therapeutic dose. These studies are intended to be performed prior to approval of the drug, of course, and most commonly after phase 2 and before phase 3 begins. Another requirement, which isn't actually part of the E14, is that the electrocardiograms from these studies are submitted in an XML format to an ECG warehouse maintained by Mortara. Now, this set of requirements has been added to and clarified over the years through the Q&A process and through direct communication from FDA. The most important change is that FDA now requires that the thorough analysis be applied to heart rate, PR, and QRS in addition to QTc. The FDA no longer requires that a clinical dose group be included in the study. That is, in most cases, a supratherapeutic dose arm is all that's needed, so long as the PK, PD measurements are appropriate to allow a good PK, PD analysis. Automation is acceptable, and it is internally validated in a thorough QT study by the presence of a positive control, usually moxifloxacin. I will touch on this topic of automation again in a moment. I skipped the moxifloxacin bullet by accident, but as most of you know, originally there was a great deal of effort in blinding moxifloxacin. That's no longer required by FDA. I personally believe it's a good idea to blind if you can. There have been recommendations offered by Zhang and Machado from FDA on how to calculate sample size, and most statisticians have accepted this approach, not because they necessarily agree, but because they know that FDA will accept it. The Bizet correction is no longer required. That's good. Multiple endpoint adjustment to control for type 1 error is required for the assay sensitivity test, and most commonly the Hochberg procedure is used. If you don't include this in your protocol, FDA will ding you and ask for it. You also need to account for possible delayed effects. Typically, this means the channel trafficking issue. and that means that you need to have at least one time point at 24 hours or later. And finally, FDA is requesting that PK/PD modeling be done in all studies. Now, let's get to the topic of what's wrong, in my opinion, with the ThoroQT study as defined by the E14. And let's do that in order to consider how we could improve these issues. The first problem I see is timing. As you can see on the graph on the left in this slide, I'm displaying curves for ECG quality in white. That basically means centralization and robust ECG methods. ECG quantity, the number of ECGs performed, in red, and the resultant information in green. And as you can see, in most cases, ECG quality is poor, the quantity is low, and the amount of information about ECG effects collected during phase 1 and phase 2 is minimal. Now, this is not the way all pharma sponsors perform their studies, but the majority do. And that means that we don't start collecting a lot of information and improving our knowledge about ECG drug effects until just before phase 3 begins. And here we see the enormous spike in all three curves, followed by a decline in ECG quantity and in ECG quality, in other words, non-centralization in phase 3, so long as the ThoroQT study was negative. Now, this timing, in my view, presents two major problems. The first one is that ECG quality, quantity, and resulting information are the lowest in phase 1, but ironically, the highest doses that are going to be seen and the most careful exploration of pharmacokinetics occurs in phase 1. So we're missing a tremendous opportunity. The second problem is that subjects are exposed to the drug in phases 1 and 2 at supratherapeutic doses. And often there are several hundred subjects and patients exposed in these phases. And my question is, aren't these people entitled to the same protection that the ThoroQT study is designed to guarantee in phase 3? Ostensibly, the TQT study is done at the beginning of phase 3 before exposure is greatly increased to the drug. But there are quite a few exposures prior to that point, and that doesn't seem rational to me. So what could we do to improve this timing issue? If you take a quick look at the graph on the left, I think you'll immediately figure out what I'm thinking here. And that is that we use rigorous electrocardiography throughout the drug development cycle. We front-load ECG acquisition, so we're getting the highest number of ECGs at the very beginning of human exposure. This centralization and high quality persists through the entire development period. But the frequency of time points and the number of replicates at each time point can be diminished as ECG information is gained, and eventually the quantity of ECGs can become quite low. The result of this approach is that high-quality ECG information is gathered during maximum dosage, and early warning of QT effects and other ECG issues is available. This improves patient and subject safety from the very beginning, and it also gives the sponsor an opportunity to get out early, so to speak, if the drug is misbehaving in relationship to ECG effects. Of course, taking this approach avoids the formal thorough QT study, and I think if you take pencil to paper, you'll find that this will reduce the cost of drug development overall. Another issue, moxifloxacin exposure. The fact is that moxifloxacin is not totally benign. It does prolong QT, and there are reports of Torsades de Pointes ventricular tachycardia occurring in subjects receiving moxifloxacin, and re-challenges have been done to pretty well confirm that moxifloxacin does do this. It's somewhat ironic that we're required to administer this drug to normal volunteers in the effort to prevent this very problem. Moxifloxacin also can cause hypersensitivity reactions. They're rare, but Stevens-Johnson syndrome and other severe hypersensitivities may occur. It also can cause deficient-associated disease after a single dose. And tendon ruptures are reported, though I suspect they would never occur after just one dose. What is the alternative for an active control? Well, first of all, I believe that a high quantity and quality of ECGs early during drug development reduces the need for an active control. Also, we now know, with several years of experience with thorough QT studies, what the methods are and where the investigative sites are that indeed can accurately measure QT and therefore don't need to be actively controlled. Also, if we maintain high-quality electrocardiography in phases 3 and 4, we reduce the chance of missed repolarization liability of a drug. So even if somehow we weren't doing a good job early on in measuring the QT, we retain the possibility of identifying problems later on by maintaining high ECG quality. And indeed, there are other very sound methods for demonstrating assay sensitivity that have been proposed. None have been used as the only method for active control as far as I know, but I may be wrong about that. Detecting the known change in QTC with positional change is one method, and using statistical methods to discern QTC precision is another approach. One other problem with the thorough QT study is the very endpoint itself, QTC. QTC prolongation has extremely low specificity for subsequent development of Torsades de Pointes. And that's a big problem. Probably the biggest problem that you can point to. It can be improved on. T-wave change in the morphology of the T-wave has higher specificity and sensitivity in animal models by a considerable degree. Currently, T-waves are assessed subjectively by eyeball by the cardiologist overreader. This subjective assessment is totally unquantified and I think less reliable than a quantification would be. So my alternative strategy here is to do other measurements, one of them being to quantify the T-wave. This might be done by T-wave segmentation, such as the T-peak to T-end measurement, or it might be done by a method that I'm going to illustrate in which the principal components or eigenvectors of the T-wave shape are quantified and then monitored over time. You're looking at an X by Y plot of T-wave morphology. When I click the Start button, you're going to see a movie of T-wave morphology change. But first I need to explain to you what's going on here. These data were generated by BioQT, which is a software utility produced by Oxford Biosignals for automated measurement of ECG intervals and T-wave morphology. And we'll be looking at T-wave morphology here. Now, this still frame shows a two-dimensional representation of the first 6 eigenvectors that describe the shape of the T-wave. These 6 dimensions or principal components are resolved to a two-dimensional depiction by a neural clustering technique, which I don't understand and which I actively avoid learning anything about. But the 2D display does make it possible for mathematical idiots like us to actually see how the T-wave morphology changes. Each heartbeat during a 24-hour Holter recording is represented in this X by Y plot by these blue dots. Each blue dot is a T-wave morphology measurement. Now, what I'm going to do is I'm going to run the movie, and you're going to see these blue dots turning yellow briefly. These yellow beats are the beats that are occurring during a 10-minute window of time. And this 10-minute window of time, in effect, slides over the data in a chronological sequence from the beginning of the Holter to the end. So you see the morphology measurement of the current beats moving through this XY plot. You will see that. We're actually only going to look at the first 9 hours of the recording. And the data will be going at a rate of 1 hour of real time per 1 second of display time. It's a fast movie. It only lasts about 20 seconds, and I'll show it twice so you can really get the idea. What you're going to see is after the first half hour of the recording, sotalol is administered at 320 milligrams orally in this particular subject, and then you begin to see dramatic T-wave change induced by the drug. At the upper left, you're going to see the Holter time displayed, and over on the right, you may see some messages, but just ignore them. We're not going to deal with them today. Now, as the movie plays, look at this pink ball right here. What that represents is an average of the 10-minute window that is currently lit up in yellow. So it gives you the average T-wave morphology at any point in time. Please don't worry about these other static green and red bubbles. We're not going to talk about them today. So I'll run the movie now and run it again. There you can see the ball is fairly static, but now it's really starting to move to different places. The drug effect is strong, and you can see huge T-wave change, and indeed you see a great deal of increase in variability of the T-wave by a greater spread of the yellow dots. I'll play this again so you can appreciate that. Dots are close together, ball's not moving much. Now there's starting to be a spread in the dots. The ball's moving further and faster. Pretty soon there's dots all over. a huge effect of sotalol on the T wave. So basically what we've done here is we've taken a rich set of quantitative data describing the shape of the T wave and turned it into a subjective moving picture. But please rest assured that these data can be used to detect and measure very precisely the extent of T wave morphology perturbation. Now let's go on to the next slide, and that's cost. The thorough QT study is an expensive study. I've seen costs ranging from $500,000 to $10 million. That $500,000 cost, by the way, did not pass through the QT IRT successfully. That study ended up costing $850,000. So it's too expensive, and it truly inhibits drug development at small biopharmas with low cash reserves. The thorough QT study can literally halt, that requirement can halt drug development. So what are some cost improvement strategies? One of them is automation. Automation of the ECG measurements. This is faster. It's much less expensive if done properly. It eliminates human error. It reduces variability, and the sample size requirement is reduced as a result. And it makes very difficult or impossible things possible. It allows a good QTCI measurement based on adequate baseline data to be calculated. It allows T-wave morphology tracking, as we've just seen, and it allows a number of other analyses that require large quantities of data that you could never ask a cardiologist to measure in any reasonable time period. And importantly, the FDA accepts these data in thorough QT studies that have active controls. There are a number of types of automation. One is the automated algorithms that you're familiar with in ECG machines, but these apply primarily to single 10-second 12-lead ECGs rather than to continuous data. There are several. I've listed 3 of the new standalone software products that are capable of measuring Holter data and making a number of useful analyses possible. So automation is one way I think we can improve cost. Another is to use purpose-built devices. I'm showing a device called the Spaulding IQ that was developed at Spaulding Clinical Research. This device was built for pharma studies, and the underlying motivation was to allow pharmaceutical companies to collect data, ECG data, inexpensively. This machine reduced the cost of manufacturing and therefore the cost of leasing the electrocardiograph to pharma sponsors. Its small size and low weight, it weighs only 3 ounces, reduces the cost of shipping. Its simplicity, you can see in the panel on the right that there are really only 3 steps to running this device, and it has only 1 button. It has 1 small LED display. It's very simple. So this reduces training costs, and it reduces human error because of its simplicity and also because it has voice biometrics instead of keyboard entry for subject ID. It enhances data quantity and quality because it provides a 5-minute recording, which allows development of a more reliable result. So using devices built for the job is another way to reduce cost. you're looking at a modified Picasso's dream. No more TQTs. The modification there is the pill bottle. That's not in the original. So in summary, the alternative strategy that I'm recommending has these elements. Front load and taper ECG acquisition to get an earlier repolarization answer. Eliminate the active control for patient safety and cost reduction. Monitor T-wave morphology to get a better assessment of risk. Automate ECG analysis to reduce costs. And use more cost-effective devices and analyses. Thanks for your attention. I believe, Rebecca, you will take over the screen display now.
Yes, that's right, Dr. Benson. Thank you, or Dr. Mason. Thank you so much for that presentation. And I'm just going to switch gears a little and come back to my screen. So as I've mentioned, thank you, Dr. Mason. I see that we have some great questions coming in, and I would encourage all members of our audience to keep them coming. We'd love to get your input and to see what you're thinking about. So at this point, I'd like to turn over the presentation to Mr. Daniel Selmas, who might want to say a few words about Dr. Charles Benson, who is our next speaker.
Sure. Thank you, Rebecca. I'm sorry?
Oh,
Dr.
sorry
Benson
about
is
that. Continue.
That's all right. Dr. Benson is a medical fellow with Eli Lilly Company. He received his bachelor's degree from MIT before attending Indiana University, where he received his MD and PhD degrees, the latter in physiology and biophysics. He then completed a residency in internal medicine at the Scripps Clinic in La Jolla, California. Dr. Benson joined Eli Lilly and Company in 2000 and is focused on early phase drug development and clinical pharmacology for the majority of his career. He's recognized for his leadership and expertise in quantitative approaches to early drug development, including work on advancing the field of prediction of arrhythmias through the measurement of the QT interval. He is currently a member of the IQ QTC working group, the OQT optimizing TQT studies through PKPD working group, and as well the ICH E14 implementation working group, as well as other efforts to further our understanding of measurement of the QT interval. Dr. Benson's topic this morning is titled Eliminating the E14 TQT. It is not if, but when. Dr. Benson has a poll question to start off the presentation, so Rebecca, could you please launch the poll question?
Thank you, Mr. Selmas. So at this point, I've just made Dr. Charles Benson a presenter, and I'm just going to let him set up. As mentioned, we will be launching a poll question very shortly. I just want to make sure that Dr. Benson is all set to go, and it looks like he is. So without further ado, let me just share the first poll question with our audience today. So while Dr. Benson sets up, our first poll question for this part of the presentation, the talk is entitled Eliminating the E14 TQT. It is not if, but when. So we wanted to hear some feedback from you on what you thought about this. So you have a few selections to choose from. The first is not in my lifetime. The second, not before I retire. The third, not in the next 10 years. The fourth option today is not in the next 5 years. And our final option for you today is in the next 5 years. So again, this is in response to the statement, eliminating the E14 TQT. It is not if, but when. So we're just going to allow a few seconds for you to think that over, and please submit your thoughts. Okay, so it looks like most of you have submitted your ideas, and I'm just going to share the responses with you right now. So in response to eliminating the E14 TQT, it is not if, but when, 4% of you thought that this wasn't going to happen in your lifetime. 5% thought that this was not going to happen before you retired. 16% of you said not in the next 10 years. 39% said not in the next 5 years. And 36% of you believed it would happen sometime in the next 5 years. So without further ado, I'd like to pass the gears over to Dr. Charles Benson, and I'd really appreciate to hear what he thinks of all this.
I thought it sounded fairly optimistic, really. A third thought in the next 5 years. I would hope so, but why don't I go through the talk, and then I'm actually going to ask you the same question again, perhaps worded slightly differently at the end, and see whether those numbers move at all. It is a provocative title with if, it's not if but when, but of course the when is not specified. It could be in our lifetimes, hopefully. But let me just show you a presentation that I did over 3 years ago, where I thought that when I was talking to the E14 group at this point, I had pretty good data to show the very same things that Dr. Mason just talked about, was that we think that phase 1 does a fairly good job. And perhaps if you do a good job in phase 1, you wouldn't need this thorough QT trial, which everyone admits has some problems.
But so Dr. Benson, sorry to interrupt. It appears
Yes.
that we're not seeing your screen at this point. I apologize. There must have been a few technical difficulties with launching those poll questions, and it must have disrupted the transition a little. So could I have request that you just share your screen? There, perfect.
How's that?
We're all set to go.
There's a button that said share, so there we go. So now you should be seeing the second slide that I have, which is basically just the title of a presentation I did to the E14 group, which had the same, again, the same approach to phase 1 being a good way to look at QT data. And what I said at that time, and I'm still saying to this day, is that we have shown that phase 1 QT data can be very sensitive to small changes. And we can use that to predict the concentrations at which the QT prolongation would be clinically relevant. But, and here's the part we still don't have agreement on, we also think that this data can be used to establish a zero or negative slope correlation of this PK/PD response. And I'll show you a few examples of that. I think you've probably seen these examples before, but let me just walk you through a couple that perhaps you haven't seen before, with this emphasis then still being on what happens with the majority of cases where you have a negative concentration QT relationship, meaning that you don't have a relationship between concentration and the QT interval, and you do power calculations to demonstrate that you could have seen a increase were one to have existed. And that's, I still think, is kind of a major sticking point right now from a regulatory standpoint. So here's an example of a first-in-man study, which was published a number of years ago by Dr. Lalonde, showing that in this case there was a substantial increase at the higher dose with this drug at the early time points with QT, and there was a significant QT PK relationship, and based off of this data and this relationship between concentration and QT prolongation, the drug was discontinued. Interestingly, this was not something that regulators intended, was to discontinue a substantial number of drugs. They, as you know, the E14 was just written to state that the TheraQT was supposed to affect the type of monitoring done in phase 3. But as us on the call know, we are using QT as a critical success factor. And even to this day, now that there are drugs on the market with increased QT prolongation, those are very few and far between, and we're still talking about killing drugs due to relatively small QT prolongations in the grand scheme of our 80 millisecond range of QT prolongation throughout a healthy person's day. So let's move on now to a positive concentration, but we have a significant difference between what we would see in a clinically relevant dose and what we would see with the QT increases. And I'll show you here a design that I did a number of years ago for an FHD. It's a fairly typical FHD design where we do a combination of crossover and dose escalation. As we usually do, we tried to push the dose as much as possible in FHD. It's one of the highlights of the advantages of doing QT measurements in the first human dose. It tends to be the trial in which you push the dose as high as it will ever be in your drug development pathway. And in this case, we pushed from 5 milligrams all the way up to 1,000 milligrams and had placebos as controls here. And this is what we showed in our by time analysis. We don't recommend doing this type of analysis. In fact, in our company, we state do not do by time analyses because of the issues with multiplicity, and you will almost always pick up one time point going higher than the other because of the smaller sample size. But this is just our routine QT monitoring that we tend to do in all of our phase 1. So there's not extra cost that's involved here. And again, an advantage of doing this type of analysis is that this is already being done, and it does not actually incur extra cost. And here's the appropriate analysis. It's the concentration QT analysis. And like much of clinical pharmacology, we think this is the more powerful analysis of looking at our PD markers, whether it's QT or other pharmacodynamic changes when compared to drug concentrations. And you can also do this with hysteresis in the model or other adjustments, but this is what we found, that the higher the concentration of the drug, the greater the prolongation in QT. And this was not unexpected, because the other thing that we always state is that we want to have a totality of evidence when we discuss the QT prolongation and the risk of QT prolongation, in that we had a mildly positive hERG channel assay with this drug, and thought that if we could get concentrations high enough, we should see some QT prolongation. And so it all makes sense. And so for us, even from a Bayesian standpoint, even though we don't have a huge sample size here, it all hangs together. And as you can see, the confidence intervals around these increases are relatively tight. And so when you look at this per dose, each of these green lines that are up and down here are the mean Cmax of the various doses from 5 milligrams way down here up through the 1,000 milligrams, which is up here. It wasn't exactly dose proportional as far as the PK wasn't exactly linear. But we could break it down as per dose based off of that concentration response model and do the more typical analysis where we look at the confidence intervals, the mean and the 90% confidence intervals around that increase at each of the doses, and show that it wasn't until we got up to 250 milligrams before we saw a upper confidence interval which exceeded that relatively arbitrary but now somewhat written in stone threshold of 10 milliseconds, which is meant to be the threshold of regulatory concern. And so therefore we argued that if we were going to go forward with a dose in clinical development which was in the lower range, and it actually did turn out that the dose that was effective in phase 2 was 5 milligrams, and we were going to go forward with 5 milligrams. We asked whether this was a large enough threshold and to not have to do a thorough QT trial. So here's the actual text of what we asked for in a thorough QT waiver that we sent to the FDA. We said, do they agree that given this positive signal, demonstrates assay sensitivity and has a significant safety margin to these planned phase 3 doses, and there's adequate evidence to grant a waiver for the conduct of a thorough QT trial? And the answer was yes. And so to my knowledge, this was the first time we'd gotten a waiver on essentially a negative QT data, because we don't believe that this was associated with any increased risk, but not having to do a thorough QT trial. And so then, again, based on the totality of evidence that includes these things we've talked about, the safety pharmacology tox phase 1 data, that the proarrhythmia risk has been adequately characterized for this 5 milligram dose. And they said, well, yes, given caveats that you don't have all of your drug-drug interaction studies done, then yes, but we certainly, if those exposures with your drug-drug interactions as well as special populations and other things exceed this threshold, then we would go back and do a thorough QT trial, and we had volunteered that. So in this case, if we have a positive response, but and then therefore the study shows that it can find a difference, then no thorough QT trial is necessary, but there are caveats around the margin of safety that we've just gone through. So let's go now to what we usually see. And so this is what we usually see, and this is another one of our phase 1 studies where we obtained an awful lot of QT data that is matched with the PK and showed that the response is essentially flat, though when you do these responses, there's almost always a very, very small negative or positive slope. In this case, it's a negative slope. And if you look across the, this case, it's a very small positive slope. If you look across, you would have to go very far out to concentrations that we would not see clinically before you got to even a 5 millisecond prolongation. And we argued that since the slope is positive but very small, the confidence interval includes 0, can be concluded that there's no clinically significant or statistically significant relationship between this concentration of drug and QT. Here's another study with drug concentrations that even were more substantial. Again, we saw a very, very small decrease, but essentially a flat concentration response. And we did confidence based off of powering this analysis and showed that the probability of there being a greater than 5 millisecond change was very, very small, less than 0.01%. And again, we argued that since the slope is very, very small, the confidence interval includes 0, can be concluded, again, there's no relationship. And the response to this was very much different. Your experience might be similar in that we were told the positive control is still needed to ensure that the study is properly designed and conducted. And in the case of these negative or neutral relationships, a positive control is needed to assure assay sensitivity. And so it got us thinking, as we have ever since the 14 came out, what exactly do they mean by assay sensitivity? We know they certainly like a positive control to help with this thing called assay sensitivity, but what exactly is it? And here's the time for our next poll question. So do I need to pause it, or you're doing it for me, right?
Yeah, sorry about that.
Okay.
There's just a little bit of a human delay on my end. So as mentioned, there's a second poll question, and I'll just set that up for you right now.
Okay, so I'll go ahead and read these. Tell me when they're on the screen, because for some reason the presenter doesn't see the polling questions. So I'm assuming that the questions are now on the screen, right? Rebecca?
Hi, Dr. Benson. Sorry, it looks like you might not be able to see these questions, but I assure you that our audience can, and they've actually
good.
Their answers.
Thanks.
So the question is, what is assay sensitivity? And you have a few choices to choose from. So the first is an ability of an assay to correctly identify a positive control. The second option is the ability of an assay to find a small difference. The third option today is the ability of the lab to accurately measure ECG Q wave to T wave. The fourth option is statistical type 2 error. That is the risk of concluding no difference. And the fifth option today is the overall performance of the assay. So again, this is our second poll question for this section. What is assay sensitivity? And it looks like most of you have already actually submitted your poll questions. So I'm just going to close this and share the responses with you right now. So in answer to the second poll question, what is assay sensitivity? 33% of you mentioned an ability of an assay to correctly identify a positive control. 37% of you thought that this meant an ability of an assay to find a small difference. 4% of you mentioned that this was the ability of a lab to accurately measure ECG Q wave to T wave. 14% of you mentioned this is a statistical type 2 error, that's the risk of concluding no difference. 13% of you thought that this meant an overall performance of the assay. So quite a few different responses there, and I'm going to leave it to Dr. Benson to help us figure out what this might mean.
Good. So it was fairly diffuse, and let me just go on to And I think that that reflects the confusion that, that we have, and that I think that the regulators also have on what exactly they mean. And so I'll tell you what the rest of the world thinks.
So, Dr. Benson, sorry to interrupt.
Oh, right, I have to share things again, don't I?
We might not be able to see your screen at this point.
Sorry.
Perfect. Okay, we're great.
Okay,
we're seeing your PowerPoint. Thanks.
And so from the rest of the scientific community, excluding QT and to a certain extent some of the clinical group, if one looks for assay sensitivity, 99% of hits on PubMed or other databases actually refer to statistical. So it's what you were taught in your statistics course, that sensitivity is statistical type 2 error. So it's a risk of concluding no difference when one actually exists. And this idea that overall performance of the assay is probably what the regulators think. In fact, when I've talked to them, they talk a lot about this overall performance and trying to get to the bottom of what they mean by the overall performance, and you list all the various pieces of that entails, whether it's actually just measuring the Q to the T wave, or whether there's some measurement of a real difference, the random variation bias, sensitivity or specificity, the stability, the drift over time. This overall performance is actually probably better described in the term validity. And so that certainly the validity of an assay is something which applies to any sort of a scientific design, whether it's clinical or non-clinical. And it's been looked at very carefully because people have been thinking through these things for hundreds of years. And there are actually books written on the topic, and the books generally discuss the term validity rather than assay sensitivity. The problem with using things like a positive control is that if the assay is valid, that is, we've used this assay before and it has worked in the past, and we've carefully designed to look for other issues like variability or bias or drift, the positive control can actually harm your assay. And I'll run you through the example. And the example is that if we ran 250 moxifloxacin studies in a row, a few of them should actually yield inconclusive results unless you have drastically overpowered your study. There should be a few false negatives or false positives, not because there's actually a problem with the study or the lab or that the truth was not found. It's just that some of these false negatives are inappropriate and then have inappropriately influenced the interpretation of the overall study, the test, and the test drug. So the overall operating characteristics of the TheraQT study have been worsened by the positive control, not improved. And then this question about is the assay sensitivity the ability to find a small difference, and that one is discussed quite a bit. And the issue here is that if, again, you have a validated way of measuring an effect across your range of interest, there's no rationale which states then you would need in your test data set to include a change in that parameter of interest for any particular subject. And so, for example, if we're looking at the assay sensitivity of LDL in a statin trial, we look to the laboratory to establish that they've got reasonable accuracy on their samples, and then we don't ask the laboratory what happens in that same subject when they go from 140 to 145. This type of a calculation is done with typical statistics, where you look at your variability in the trial, you ask whether that you are powered, given that variability, to find a difference where one to exist. And so I think that what we need to do then is focus on exactly what it is what they're interested in. And so most of the time we state that we're interested in this trial to demonstrate that our effect is lower than a threshold value, in this case it's 10 milliseconds. And we can do that by looking at our type 2 error, our false negative rate. We can use traditional statistics to do a power calculation to ask whether we were powered to find a difference of 10 milliseconds were one to exist. And if we are, which we think that we did show in our phase 1 negative studies, then we should be okay. However, it's also clear that the regulators, and perhaps others in the QT field, mean more than that and try to apply the term assay sensitivity to describe the overall performance of the assay. And that's problematic, because if they're asking for us for something other than the traditional use of assay sensitivity across the scientific community, we should specify what it is exactly that we're worried about. And when I've talked to the regulators, different regulators are interested in different things. So some of them actually think that we have a problem with ECG measurement, that certainly in the past there have been problems measuring the Q wave to the T wave. However, that field, like many, have matured, and we think that we do a pretty good job, especially some of the automated systems have been shown to reliably measure the Q to the T. The other things like random variation bias or sensitivity or drift can be accounted for in your study design. And if they tell us exactly what it is that they're interested in from a overall performance standpoint, then we can design the study to address those particular needs. So I'll move on from validity because it is still something that we need to address. But one approach has been that we look at a prospective study on these early clinicals to try to establish that they are a valid system. And what happened was that we were trying to convince regulators that we do a good job in this phase 1. And so what they did was they went and pulled all of their TheraQT trials, which is now over 250 that they had in the FDA database, and compare them to what early phase data they had for those trials, which were positive. And let me show you the results here. So we've already talked about this. TheraQT studies certainly require a substantial outlay. We might be outliers, but Lilly has now run a $16 million TheraQT trial and a $14 million one. But that aside, can we use these non-clinical and early clinical data to replace a TheraQT trial? They looked at their 55 out of 250 TheraQT trials that were positive. Of the 55, 31 were performed after January 1st, really after the E14 came out. Of those 31, there was 17 of these was early clinical data on QT measurements. Of those 17, 6 of them were found by the sponsor to have a positive signal in those early phase clinical trials. 6 out of 17 is fairly poor sensitivity. However, none of them included a concentration response analysis, which we think is the most important analysis in phase 1. And so we say that this is probably due to not using our kind of modern approaches to QT. We think we do a better job measuring QT early on these days. And they said, well, okay, fine, prove it. And so we made this proposal. And so we proposed to do a prospective early clinical QT study with drugs which do prolong QT. And we run a study very similar to what we do in phase 1 and show that we don't miss any of these signals. And we asked, well, in order to do this, it's going to take quite a bit of money and a lot of companies working together. We'd have to have a pretty big carrot. And they said, well, you wouldn't have to do TheraQT trials if you would have confirmed that you can find these signals. And we thought that that was a good carrot. So there's some limitations to this approach in that this study that we're proposing needs to be reasonably small, and we won't do the full SAD type of an approach. We won't use all the dose cohorts that we typically have in a SAD, so it's reasonably conservative. We would use single doses and prove then our ability to look at QT after phase 1. And then if we do our studies from here forward in drug development, similar to what we did in this study, then we would get a TheraQT waiver. Our design here is a crossover study using 8 to 12 per cohort. We have to look very carefully at the power of this, but we think that it will be good if we use 2 consecutive days with a kind of a clinical dose followed by a maximum tolerated dose with a placebo arm and a somewhat blinded study. Here's the actual design with 4-period crossover with 3 active drugs and 1 placebo. And then the test drugs will likely be able to be used in healthy volunteers. And then we would have this study again with a single dose with at maximum tolerated dose, which is at least representative and probably higher than what would occur at steady state. We wouldn't include drugs with a negative TheraQT besides the placebo due to sample size limitations and this focus on sensitivity, a false negative rate. And then, again, the conduct would be using what we think are state-of-the-art clinical conduct with the emphasis on reducing autonomic tone, heart rate variability. We probably use a continuous ECG recording and of course PK sampling to support the concentration response analysis. We would do a pre-dose baseline before each crossover period rather than a full baseline day, and then do extractions which are verified for stability of heart rate. Moxifloxacin will be used as one of the test drugs, but it will not be necessary in routine practice after we show the validity of this assay and this approach. Perhaps we can look at some of these other pieces that could be thought of as helping with validity, such as intrinsic variance or heart rate variability as supportive data. From our ECG assessment, we'll likely use centralized analysis with replicate extractions from a period of, again, stable heart rate. We are probably going to use multiple core labs to show that there are multiple ways that we can measure these QT intervals. Our primary endpoint will be a concentration effect model, such as what we showed earlier in this presentation, and we'll use some central statistics, but not use as a success criterion. And so if this positive outcome of this proposed phase 1 study correctly predicts the positive TheraQT study outcome for all of the drugs selected by the FDA, then a negative outcome of future routine phase 1 studies conducted in the same robust fashion would support the TheraQT waiver. And again, what we're trying to show is that even with small-ish sample sizes in phase 1, using techniques that are reasonably common in most phase 1 studies, that we can find these small changes. And so overall, we thought that an early phase QT, if significantly positive or negative slope, can be used to obtain a waiver, and a proposal is being made through the CSRC that clinical pharmacology study, if successful, will convince the FDA that TheraQT trials are no longer necessary. And so at this point, I put up the final polling question again and see if some of the answers have changed, given that I've changed the question slightly in that instead of stating eliminating the QT if not, it's not if, but when, I say obtaining a waiver for the TheraQT is possible with careful phase 1 QT data collection, but that I have the same answers, not in my lifetime, not before I retire, not in the next 10 years, not in the next 5 years, but in the next 5 years is E. So if you can answer those questions, answer that question, and then we'll talk about it.
Thank you, Dr. Benson. And while our audience is taking a moment to consider those questions, I'd like to also bring back Dr. Mason and thank him again for his insightful presentation. So again, the question that's appearing on your screen right now is, obtaining a waiver for the TQT is possible with careful phase 1 QT data collection. And your options are not in my lifetime, not before I retire, not in the next 10 years, not in the next 5 years, or in the next 5 years. So it looks like most of you have had a chance to consider that question. So I'm just going to close it and share the results with you right now. So in answer to that question, obtaining a waiver for the TQT is possible with careful phase 1 QT data collection. 2% mentioned not in my lifetime, 1% mentioned not before I retire, 3% mentioned not in the next 10 years, 21% mentioned not in the next 5 years, and 72% mentioned in the next 5 years. So that's a very interesting difference from the first poll question that we launched earlier in this webinar. So at this point, I'd like to turn the presentation over to Mr. Selmas, who will be moderating our Q&A session. So, Mr. Selmas, would you mind saying a few words and taking it from here?
Sure. Thank you very much, Rebecca. Thank you to all of you who have posed questions throughout, and again, thank you for kind of holding and waiting on those replies at least until the end here. We just felt it important to make sure that Dr. Benson and Dr. Mason both got all of their content out there. But with that being said, I think we can spend a little bit of time perhaps going through some of the questions here. And I have about 7 or 8 of them that I think we can start out with here, some of which may have been covered to a degree in Dr. Benson's presentation, but I think Dr. Benson and Dr. Mason, please feel free to chime in as I bring these questions up here. So the first one was, you're concerned about the timing of thorough ECG evaluation during the development cycle is certainly understandable for the reasons given. Unfortunately, the majority of early phase 1 trials are small N studies. How can you be sure the ECG results are reliable with such small sample sizes?
Dan, are you going to assign the responder here, or do you want us to just?
Sure, Jay. So Jay, if you'd like to take that one, I think you were looking at the first couple there that perhaps you could.
Yeah,
ask.
I think the first several are ones that I think I can answer. I think this is a good point. We have small numbers in phase 1 trials, but I think there are two saving graces here. One is that each study has a small number, but during phase 1 and phase 2, several studies are done, and generally you've got well over 50 subjects in whom you can evaluate ECG change. In addition, as Dr. Benson pointed out, one can use PK/PD assessments in relatively small numbers successfully. So I agree with the issue. We have small numbers, but I think it can be overcome. Dan, the second question, well, I think I can paraphrase it so that you don't have to state it. This second question is a good point. The questioner indicates that 50% of drugs that enter phase 1 are discarded, and usually in phase 1 or phase 2. And if we're going to follow the strategy of front-loading the ECG work, we're going to end up paying that price in quite a few drugs that don't make the grade. And I agree with that. But yeah, I think if you look at a pharmaceutical company as a whole, a company that is developing many drugs, or if you look at the industry as a whole, developing many drugs, indeed, you'll lose some money on some of them early, but you're going to get that back and more by avoiding doing thorough QT studies later. And you're also going to abandon some drugs appropriately earlier. So I think the net is in favor of the pharmaceutical companies. Dan,
add to that?
Yes, please.
And just to emphasize that, again, if we do this concentration response, I think, appropriately, we are not going substantially above and beyond what the current practice would be. So currently in our phase 1, especially small molecule studies, we do these concentration response analyses on QT currently. And what we're asking for here is really not above and beyond our current approach. All we're doing is we're asking that that current approach be then validated to substitute for a thorough QT study when you incorporate the non-clinical data as well.
Beautiful. I think the next two, Dr. Mason, go back to your video. One of the questions was, please clarify what the axes in the graph are. And then the second one says, your video highlighted the T-wave effect. What was happening to the QTCF over that same time interval?
Thanks, Dan. I can answer that in 1 minute if you can give me screen control, and I can show that that movie. Show my screen. Okay. Let's go to the movie and, well, all right. The first, what is the axis? Well, that's that sort of magical arithmetic I was talking about. So I can only tell you in a subjective way what the X and Y axes are. You can think of them an easy way to think about it is that one axis is the amplitude of the T wave, and the other axis is the duration of the T wave. That's not, in fact, what they are. This is a mystical arithmetic way of taking 6 dimensions and resolving it to 2 dimensions, so you can, so a normal human can look at it. So again, if you think about it as size and duration, that might help you with that question. Now, the second question is what's going on with QTCF while the T wave change is occurring? And so I want to draw your attention to the messages that are going to appear up here in the upper right. One tells you when maximum QTCF change has occurred, and the other when maximum T wave change has occurred. And what's interesting here is that the two are dissociated. And the T wave maximizes later than the QTCF. QTCF went up about a mean of, honestly, I can't tell you what it was in this one subject. In this study, the mean change was about 30 milliseconds, so substantial. But let me run this, and you'll see how the QTCF and the T wave morphology are dissociated. Well, that's interesting. I'll get it right. Hang on. There we go. Look up here for the messages. Maximum QT change. Maximum morphology change. They're dissociated. They were dissociated by about 90 minutes. So interesting, and I've speculated that this dissociation is an indicator of badness because the QTC change, the T wave morphology change seen for moxifloxacin and in fact for no drug, the changes are smaller, but the T wave change and the QTCF change coincide almost perfectly. And of course, no drug and moxifloxacin are much safer than sotalol, much less arrhythmogenic.
Great.
Thanks, Dan.
You bet. Thank you, Jay. One quick question. We want to launch one more poll question that an attendee had sent in. We thought it'd be really good to ask the group about it. So I'll turn it to Rebecca to do that quickly, and then we'll have 2 more questions.
Great. Thanks so much, Mr. Selman. So what the attendees should be seeing on their screen right now is our last quick poll question of this webinar. How many attendees have worked on a drug that was killed by a positive QT result? So you have 2 choices here, either yes or no. And I see a lot of you are busy submitting your responses and thinking that over. So again, the question is, how many attendees have worked on a drug that was killed by a positive QT result? And you can pick either yes or no. So
it might help to, can you hear me? It might help to clarify this. Are we talking about a QT finding at any time during the drug's development, or are we talking about a thorough QT study finding?
Yeah, so we didn't specify, and it looks like most of the responses are already in.
Okay.
yes, unfortunately, that's right. Our attendees have already jumped the gun and answered in their own way. So it appears that 32% answered yes, and while 68% answered no. So I'll leave it to the team to figure out maybe what that means.
Sure. So, Dr. Mason or Dr. Benson, is that alarming to you that that a third of the attendees here have had a drug killed because of it, or perhaps that's why they were very interested in this session?
Good point. From my perspective, this would be much easier to address if we knew not just the number of attendees, but the number of drugs that were that the attendees worked on. So, you know, we've got a third of the attendees with a drug getting killed, but maybe it was only 1 out of 20 drugs they've worked on. It's a little hard to understand what the number means.
Sure, we'd almost need a statistician for that.
Yeah.
I'd still emphasize that this is alarming to the FDA, however, because again, this was not their intention with the E14 to go out and kill drugs with it. And they're very worried that good drugs have been stopped for QT prolongation, which is not arrhythmogenic. And so I think continued work on arrhythmogenicity, as well as improved understanding of perhaps false positive findings on QT studies would improve our overall success in getting drugs to patients.
Yeah, that's a very good point. I certainly agree.
Another quick question, I believe, directing to Dr. Benson. For the plots of the QT versus plasma concentrations, were those Cmax-based assessments, and if so, isn't there still a chance of missing a signal that may have been time or hysteresis-based?
Right. So these were all of our concentrations, and it's appropriate to include all of your concentrations in the model if you do an appropriate check for hysteresis, because we certainly think that there is some delta between your plasma concentration, which you're measuring, of your drug, and your QT prolongation, which you're measuring based off of the concentration of the drug, perhaps at the level of myocardium. However, those type of adjustments can be made with a typical established method of adjusting for hysteresis within the model. So the first thing you do is you check for hysteresis, and if present, you make the appropriate adjustment. If not, you don't have to.
Excellent. And then we have one last question of given your recommendation to conduct single-dose early phase 1 design, what do you propose for long half-life compounds that require multiple doses to achieve steady-state concentrations?
Yeah, certainly that can be problematic. What we'd like to hope is that if you've got a large enough margin of safety and not have a significant tachyphylaxis to issues regarding tolerability, that you can use a loading dose. So for example, if our drug that I mentioned in my phase 1 study had a substantially long half-life, we could have achieved those concentrations with our 1,000 milligram dose, given that we're going into phase 3 with a 5 milligram dose. That perhaps is an extreme example, but that's, of course, what we would hope. Alternatively, you would just have to bite the bullet and do a longer study than a single-dose study if there are, and those are more problematic from a variability standpoint, but you can't account for that and potentially generate the same data off of your multiple-dose study rather than just off of your single-dose study. And so can I just answer one more question that I saw come up? I don't think I was explicit enough in discussing this prospective study. That it's that I'm a member of, I'm employed by Lilly, but this prospective study that we're talking about running is not through Lilly. It's through a consortium. It's the CSRC that many of you are familiar with, and that will be running it through that consortium, which is a combination of multiple industry members and academia, Duke specifically, but other academic institutions are also a member of that consortium, and actually the FDA as well. So all three of us will be working together. How exactly that's going to be financed still needs to be worked out, and of course the devil's in the details. But I think everyone's very excited about this potential new data set, and hopefully we'll be able to work out the finances of this, because we think that this study will likely be a drop in the bucket compared to the overall expense of the industry on thorough QT studies.
Thank you, Dr. Benson. Thank you, Dr. Mason. So we're coming up on the end of our time here, but I just wanted to, before turning it over, just thank everyone for joining us. I hope you really got something out of this session, and we'd love to continue discussions with anyone that's on this webinar moving forward here. Thank you.
Thank you, Mr. Selmas. And on that note, I'd also like to thank everyone for participating in today's conference. We got some great questions in, and some great poll questions were asked. So a notice to all our attendees, you will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. At this point, I'd like to thank our speakers, Dr. Jay Mason, as well as Dr. Charles Benson, as well as our moderator today, Mr. Daniel Selmas. We hope you found this conference informative, and have a great day.